Hey, Hammy here with video number 4 on proteins from section 3.4 from chapter 3. Okay, when we look at sort of the monomer of proteins, we're talking about amino acids, and we'll take a closer look at that structure here in a little bit in a future slide. Those amino acids are linked together by peptide bonds, which is a covalent bond between the two amino acids. Proteins are very, very numerous in living things. If you take any sort of living tissue, about 50% of that dry weight is proteins. Proteins have many important functions. There's several 100,000 that are used in living things that have been identified, and could be a lot more. Two or more amino acids joined together are called a peptide. And when you start stringing a whole bunch of amino acids together, you get a polypeptide. When that polypeptide kind of assumes its final shape, we call it a protein. Or certain proteins might actually be several polypeptides that are joined together that are in its kind of final functioning form. There are 6 main functions of proteins, so you can see their role in cells and living things is quite diverse. First off, number 1, metabolism. Remember, that's the sum of all chemical reactions happening in the cell. Well, a lot of proteins are enzymes. Enzymes are protein catalysts in living things. And you need an enzyme for just about every single reaction that's taken place in your body, in your cells. They bring 2 substrates together and help cause anabolic reactions building. They will also cleave bonds and do catabolic reactions, which break them apart. Number 2, support. Support tissues. Proteins have a structural function. For example, the keratin in your hair and nails, collagen in your skin, that give them structural, kind of help support other tissues. Number 3, transport. They help move things. They move things in and out of the cell. Channel and carrier proteins allow things to go through the cell membrane. There's actually little pumps that will pump ions in or against a concentration gradient. Also transport things throughout our body. For example, hemoglobin is a protein that carries oxygen out to our tissues and cells. So in and out of cells, also moving things through living things. Number 4, defense. Antibodies, which is used by our immune system, help bind to antigens of cells, invaders, and help neutralize them and keep them from damaging our tissues. 5, regulation. Some hormones are proteins that have regulatory functions, like insulin is a protein hormone that controls our blood sugar levels. And number 6, motion. We learned about this in Bio 2, the actin and myosin, that have the little bridge buoys that help the sliding filament model that slide those things past each other. So they can be used in muscles to help the organism move. There's also little motor proteins within the cell that help move transport vesicles to the different organelles within the cell itself. There are 20 different amino acids that are very common and used in most living things. We can make all those thousands of proteins from these 20 different amino acids. When you look at their structure, their base structure, remember we often learn this as North Carolina Cougars, NCC. This is the central carbon right here that just has a hydrogen off of one side. Off of the left-hand side here is an amino group with a nitrogen and 2 hydrogens. Again, if we remember our functional groups from last chapter. And over here, it's not labeled, but they often call it an acidic group, or this would be a carboxylic acid. So you have an amino group and an acidic group, amino acid, Wow, clever in the naming there, right? The carboxylic acid has the double bonded oxygen and then also an OH group. Off the bottom of that carbon is an R group. This just is like, I like to think of it as the remainder of the molecule. This can vary from a hydrogen to these very complex ring structures. Here are some of the common amino acids that we find in living tissues. And notice that they've highlighted the R groups, like right here. Okay, and these R groups give them very different properties. Okay, so these ones up here, some have nonpolar or hydrophobic R groups. These down here have hydrophilic or water-loving R groups. And this will very much help determine that final shape of the protein, depending on how these interact when you start linking all these amino acids together. Down here is ionized R groups. And again, you can see that they go from very simple, here's just a CH2 with an OH, so a carbon with a hydrogen, and an OH group, to these very complex ring structures like you find down here. Or here's a nitrogenous ring on histidine down here. Again, a review like we've done with the other biomolecules. How do we link these monomers together? You got her. Dehydration, synthesis, or degradation would be hydrolysis. And so I've just kind of split this and blown this up to make this bigger. Here's amino acid number 1. Here's amino acid number 2. Okay, we take the OH off the acidic group over here and the H off the amino group, off the nitrogen here. So again, we take out an HOH or a water, an H2O, dehydrate, and then that bond, you can see down here, that bond right here is the peptide bond that links the two together. Okay, that links the two together. And then for hydrolysis, it just goes back the other way. You put the water in, and it will break them apart. Now, something I want you to notice is that in this group here, this NH group has a weak affinity for electrons, whereas this oxygen, double bonded oxygen, has a high affinity. So you can actually get some polarity going on here, where this oxygen side here will be slightly negative, and this hydrogen down here will be slightly positive. That'll be important when we start talking about structure of proteins, because when these things start linking together, you can actually get some hydrogen bonding that will occur because of this little polarity and unequal sharing of electrons that's going on here in this peptide bond. When we talk about structure or shape of proteins, that is very, very important. Proteins must have a proper shape. I'll refer to this idea of F cubed, okay, which means form, fits, function, okay? And that happens a lot in biological systems, where the form or the shape of the protein fits its function. So if shape is very important, if it loses that shape, we say it's denatured. So when an enzyme is denatured because of a change in pH or too high of a temperature, can disrupt those bonding patterns within that chain of amino acids, then it loses its shape, and then it doesn't work anymore, doesn't work properly, or doesn't work well. And so we say that the protein is denatured. There are 4 levels of protein structure, and we'll look a little bit closer at this on the next slide. The first level is just the primary structure, and on the next slide we'll look at actual pictures of these. But the primary level of a protein is just a linear sequence of amino acids. So you just have amino acid, peptide bond, amino acid, amino acid, amino acid, amino acid, amino acid. It's just a link, like I think of like little pop beads that you pop together, and you just make a long chain of them. Hundreds of thousands of different polypeptides can be made from 20 amino acids. We say, how is that possible? Well, how many letters in the English alphabet? You got 26 letters. how many words that can equal how many words in the English language? A lot, right? Because you can use them in various number and sequence. And so it's the same way with amino acids. We can take those 20 amino acids, use different amounts of them, and put them in different sequences, and we can get a whole bunch of different kinds of proteins. Changing that sequence of amino acids can produce different proteins, and that is all determined by our genetic code, which we'll talk later about this year, how we have that triplet code, and each of those triplet codes, 3 nucleotides will determine what amino acid is placed next. The secondary level is where this chain of amino acids, Linus Pauling did a lot of work with this, and they're just called alpha and beta. Alpha helices, because alpha, first letter of the Greek alphabet, he found that one first. And it's just where the chain of amino acids just start to coil up. And they get some hydrogen bonding and stuff happening between the R groups of the amino acids. They also found beta, or pleated sheets, where they tend to do more of a zigzag like this instead of coiling. Okay. So in one amino acid, you might have a helice, and then it might go into a beta sheet, then it might go into a helice, and then another beta sheet. So it can kind of, different parts of that polypeptide chain can have both of these, which leads to number 3, the tertiary level, where it starts to kind of coil and clump up together. A lot of that is based on the hydrophilic and hydrophobic portions. The hydrophobic portions of the protein will want to be away from the water, so they will orient or fold themselves towards the inside of the protein. And then the hydrophilic parts of the protein will be on the outside, because the water in the cytoplasm or the organelle, that interstitial fluid or cytoplasmic fluid, is mostly water. So the hydrophobic parts will clump up in the center. And then you start to get other interactions of covalent bonding and ionic bonding. You get some disulfide bonds, which help hold that structure together. And they sort of clump up, and we say a lot of proteins are globular, because they look like globs. They sort of clump together. Quaternary level, the last level, is just when you have more than one polypeptide. For example, hemoglobin, carrying oxygen in our blood, is 4 polypeptides joined together. And so that would be the quaternary level, is when you get all the polypeptides together to form that final protein shape that has a certain function. This slide is just showing more of a visual of those 4 levels. With right here being primary structure. So again, you just see like a long snake or a long chain of amino acids. Okay. Number 2, the secondary structure. We've got 2 of them. Okay. Here's our alpha helix, where you can see it's just coiled up. Notice the hydrogen bonding here between the remember I said how in that between the carboxylic, the acidic group, it's an unequal sharing. So here's that oxygen and hydrogen, that's slightly positive, slightly negative kind of hydrogen bonding up. Same thing in pleated sheets, where you see it's more of this bent zigzag back and forth. Number 3, the tertiary structure, where you start to get things like disulfide bonds connecting them. But here you see some helices, and you also see some beta sheets, and they all start to coil and clump together. The quaternary structure, they just use different colors. Hemoglobin, there's 2 alpha proteins and 2 beta proteins. So there's polypeptides. So there's actually 1, 2, 3, 4 polypeptides all joined together to make that one hemoglobin protein. Again, some more visuals of how proteins can actually affect structure. A lot of fibrous proteins, for example, the keratin in hair, is mostly helices. it's all helices, and it's just helices, helices, helices. But we can actually use chemicals like a perm or heat. If you use a curling iron or a straightening iron, and you can actually change some of those bonding patterns between the amino acids, between the polypeptides, to make your hair straight or curly. Sometimes water or humidity. You ever say, "Oh, it's so humid, I'm having a bad hair day." I have those all the time, right? The humidity, water vapor, can actually interfere with this as well. So that's why when it's really humid, it messes up with that bonding in your hair, and it gets more frizzy, or it gets kind of more volume. Spider web, as an example of more beta or pleated sheets that just kind of coil back and forth on each other, and actually very, very strong structure. Finally, is just some of the research that they've done with proteins that don't work correctly. Two things. They've found chaperone proteins, like a chaperone goes along to make sure everyone's behaving right on a trip with kids on a field trip. Chaperone proteins help proteins fold into their normal shape. They found that there's these helpers that sort of take the proteins and help them get into their final shape. Defects in that can have some bad consequences. In humans, they're looking at the role of chaperone proteins in Alzheimer's disease, also CF or cystic fibrosis, and the consequences that has on the human body, on the human system. The second one would be prions. The TSCs, the transmissible spongiform encephalopathies, are misfolded proteins causing brain disease, things like mad cow disease, as an example of a TSC. Prions are misfolded proteins, and they affect the folding of other similar proteins. And this messes up things in the nervous system. So it's not actually a bacteria or a virus that's causing these, but actually a protein that is causing this disease. And that really has threw scientists and immunologists and stuff off at the beginning. So these are just some examples of kind of proteins gone rogue that are not working correctly. Okay, so this kind of finishes up our section on proteins. There will be one final video for this chapter in which we will talk about nucleic acids.